This is Dr. Ippolito, and this is the genetics lecture for BIOL101. This will cover everything the book talks about from Mendel to Thomas Morgan and then beyond that. So let's get started. All right, so first of all, you can see these silly pigs here. I have to change that. Breeders used to believe that when you took a skinny pig and you crossed it with a medium-sized pig, you'd eventually get fatter pigs. And so the idea of blending was the most popular theory up through the Middle Ages and actually into the late 1800s. So the idea here is that offspring are a blend of their parents. Okay, so the way this theory goes though, and here's the prediction it would make, is that as you take these different parents and you cross them and you get blends, over time you get everybody looking the same. Okay, the parental traits would be lost to time. The problem is the theory doesn't fit the data. Okay, so for example, if you take some really black coffee and some really white milk and you mix them together, you get, varying shades of, beige. Now imagine trying to mix these beiges together to restore and get the black. You can't, right? And so that was the biggest fallacy of the blending theory. Okay, so as we said, this isn't what we see for many traits. So the fact that even today, after humans have been around for a really long time, we still see these variations from dark to white, for example, in skin color. Or pretty much any variation, right? Okay, so you can see, so she's got wavy hair, he's got straight hair, she has really curly hair, right? So there's straight and curly hair, there's wavy hair. Things don't blend over time, okay? And we even see that some traits skip a generation, right? So think of things like malpattern baldness, a mother's your mom's dad, your grandpa on your mom's side, if he was bald and you're a male, there's a chance that you'll be bald. Okay, now the problem was that blending was a big challenge for evolution theory. Okay, because for Darwin's theory to make sense, there had to be some kind of specific mechanism for variations to be conserved over time. And despite the fact that this was the most popular theory, blending, it didn't support Darwin's theory. So combine that with a popular biblical notion that species were specifically created and did not change, the acceptance of Darwin's work hit a really significant roadblock. Okay, now in general, when the population believes something, okay, the burden of proof is on the person coming up with the new idea. And biology, really up until the early 1900s, was mostly an observational science, in the same way that astronomy is still today, right? We don't do experiments blowing up stars and then seeing what happens. And so the notion of experimental biology was still pretty new and rare in Darwin's time, in about the mid 1800s. And the most people that dealt with breeding weren't scientists, they were breeders. And breeders did tend to see blending pretty frequently. So why do you think that is? Okay, so most traits are due to the activity of multiple genes and also environmental contributions. So it turns out that even though traits as we know today are controlled by genes, the outward appearance of a phenotype, or what we look like, tends to be, observationally, it looks kind of like a blend for many traits, not all traits though. All right, so an analogy here would be like paint pigments, right? These are actually single molecules, right? So if you were able to zoom in on paint, you'd see individual, particles of paint, and each individual particle would be a specific color, right? So this particle would be green, and this particle might be blue. Or let's say that we have yellow and blue. Okay, we have yellow and blue paint, and of course, if you guys remember your art lessons, yellow and blue makes green, right? And so if we zoomed into this green though, we would see lots and lots of individual pigment colors that are individually yellow. Oops. Yellow. Oh boy. Or blue. Okay. So the idea here is these individual dots could represent like genes, for example. All right, so as I said, when the theory is contested, the burden of proof's on those that believe in the new theory. So for example, we've gone through very significant changes over the last few centuries, right? We used to believe the Earth was flat based on our observations. We used to believe the Earth was young based on what the Bible tells us and the fact that Western civilization is generally built upon biblical notions. We used to believe that the sun travels around the Earth, okay? And so you have the geocentric versus the heliocentric model. And that lead can be turned into gold, so we had alchemy, and of course traits are inherited as a blend of their parents. All right, so for example, inheritance is nothing more than the blending of parental traits. Offspring are the average of the parents. Well, then why are traits that vanished occasionally seen again? The mechanism is unstable, but generally holds true. Well, I believe there are certain pure factors that do not get diluted, but instead passed down passed down to offspring in their original form. Prove it, right? And so this is what Mendel set out to do. Now, Mendel was born in the early 1800s in Austria-Hungary, and I'll make a long story short here. He eventually joined an abbey and studied genetics, among other things. He eventually became a teacher as well, and eventually the head abbot of the monastery. And when he passed away, his work, because it was so complicated, passed away with him. And it wasn't until the early 1900s that he was rediscovered. Okay, now his success was from his deliberate, carefully selected scientific toolbox, his P's, Pisum sativum. He selected simple traits, and he most likely did a lot of research before arriving at his P's and the 22 traits. Okay, so the requirements for his experimental plan was the substance had to be quick and easy to grow. It had to be able to cross-pollinate and self-pollinate. We'll explain that in a bit. And it contains true breeding traits. Okay, so there's Mendel. In a picture back in the 1800s, you can see he's focused so much on his P's that he wasn't even paying attention to the picture. Okay. So what made the P's ideal? Well, so first of all, there were many variable traits within the species. Mendel could actually go to market and find lots of varieties of different peas. Normally, the pea itself is self-fertilizing. The plant itself is a hermaphrodite. What that means is the female and the male parts are within the same individual. I'll show you a diagram in a second. They're easy to breed, and they're able to establish what are called true breeding lines. A true breeding line would be a plant that is able to pass a trait on to all of its offspring, and those individuals, the progeny, would be able to pass that trait on for many generations without alteration, assuming no mutation, of course. So the flowers were also large enough to make crosses really easy when they wanted to be, and so we were able to both do cross-fertilization and self-pollination. All right, so here we go. We see, here's the anatomy of the pea. So within the flower itself, you can see that there's something called the stamen. Here, let me change the color of my pointer here. We got some red, there we go. Okay, so we have the stamen. The stamen are these yellow pieces, okay? And so there's the filament, and there's the anther. This is where pollen comes from, on the anther. Okay, now the green portion here, that's the carpel. That whole thing is called the carpel. And the carpel is tipped by something called the stigma. The stigma is sticky, and that's where pollen grains would come and stick to it. Okay, and then there's this long area here called the style. And then this here is the ovary. And within the ovary are individual ovules. Okay, these are gametes. And each one of these is its own individual. Okay, so you can take a flower and you can remove the stamen by cutting them off with a pair of scissors, and you've essentially made the plant into a female. And then isolate it, and then what you can do is you can take a male plant and you can take the pollen from it, and using a paintbrush transfer the pollen onto the plant stigma. So you can do what's called a hybridization or a cross-pollination. Okay, so basically you can either have one plant that allows itself to be self-pollinated, Okay, and so that'll be represented in the future slides by a circle that looks like a P and then an arrow that does this. So that's self-pollination, or you can allow for cross-pollination or hybridization. Those are interchangeable. Okay, this is the slide we're going to come back to a couple times. It's a little overwhelming, so let's just go through this. Here are the individual traits that Mendel observed in peas, that he, rather I should say, selected for his experiments. The one we're really going to deal with right now is seed color, okay, and then eventually we're going to deal with stem length. So for seed color, we're going to talk about yellow and green peas. Don't worry about any of this right now. Don't worry about any of this. Just focus on the fact that we're going to deal with plants that can be either yellow or green. Okay, so we're going to take a green pea plant and we're going to allow it to self-pollinate, and then in subsequent generations we're going to make sure that it's always green. And we're going to do the same for yellow. Okay, and so we can call these the true breeding varieties. Okay, now understand what we're thinking about here when we talk about reproduction and going to the next generation from P to P. Okay, the key is that each individual will make gametes, right? So just remember that. We're going to have to deal with gametes. All right, so when we do the cross, the idea is if this is the original P, or the P generation, the parent generation, the first generation of offspring we call the first filial generation, or just the F1s. Okay, and then we can name every generation after that a filial generation, F2, F3, etc. Okay, all derived from a parental stream. All right, so the first cross Mendel did was he took a female, or mom, that was yellow, and allowed it to hybridize, or cross-pollinate, from a pollen from dad. That was green. And it turned out the first generation was all bright yellow, just as yellow as mom. It was not a mixture of blending. So this was the first refutation of the blending theory. And just to be sure it wasn't related to the gender, we had a yellow dad and a green mom, and we had the same results. Okay, so together, this kind of cross where you have both parents swapping the phenotypes, that's called a reciprocal, oops, whoa, let's go back there. Let me try writing that somewhere else. Reciprocal cross. Okay? All right, so because yellow came out of the F1 generation, we're going to call that the dominant phenotype. And the hidden phenotype, the green one, that we can't see in the F1, we're going to call that the recessive phenotype. Okay, so what happens now if we allow that F1 generation to self-pollinate and get the F2 generation? Okay, so if the blending theory was true, we'd always get yellow, right? The green trait would be lost to time. It would be gone forever. However, if there was some kind of pure unit for green, some factor for green, we would expect to see it again. Okay, and sure enough, when we allow that F1 generation to pollinate, okay, if we think about 4 individual offspring, right? So we allow the F1 to self-pollinate, and then we count all the different offspring that come out of it, all the F2s. It turns out that 75% of the time we get a yellow pea, but then 25% of the time we get a green pea. No matter how many we count, we always get a 3-to-1 ratio. So we would call this the phenotypic ratio. Okay, phenotypic ratio of 3-to-1, right? And again, remember, phenotype is the outward appearance of the gene, while the genotype is the actual genes, which we haven't talked about yet. All right, so there it is. The vanished trait reappears in the F2, and we call this the recessive trait, and we can think of this as the recessive phenotype, green. We can also now start talking about the actual genes involved. Okay, so we would say that the plant that's green has what we call the recessive allele. We'll talk about alleles a bit later. All right, so Mendel, doing this work, established what we call the first law of genetics, or the law of segregation. And here's how it goes. So for each trait, such as pea color, there are 2 factors present in each individual. And by individual, we're talking about the adult organism, the adult diploid organism. Okay, so this factor is some kind of physical thing, which we understand as genes, but Mendel had no idea. Okay, chromosomes weren't even discovered by the time he was coming up with this concept. So these factors within the individual were separated. These 2 factors were separated when gametes are formed. Okay, so the gametes themselves only have 1 factor per trait. Where have we seen this before? If you think about meiosis and you think about a pair of chromosomes, right? Okay, we'll get back to this in a second. So when the gametes come back together again, so you have two gametes that go through fertilization and you get a zygote, that new individual has two factors again. Okay, so the parent has two for each trait. The parent makes gametes that have only one. Okay, and so of course we have two parents that each make gametes. Oops, I just said that wrong. Let me, how can I erase that? Is there an eraser here? I think there is. Yeah, there it is. Okay, let's just do. Okay, let's go back in here. Where's my pen? Okay, so, okay. So these two represent how many factors? So there's two factors for each trait in the adult, right? And so let's say this is mom and this is dad. Okay, and so these are the gametes. And the gametes only have one factor for each trait. And so upon fertilization, when you take that egg and sperm and you get a zygote, the zygote once again has two factors. Okay, this completely mirrors what we learned in meiosis. Okay, so in meiosis, you have a cell that has some number of homologs. Okay, so let's just say for our purposes, we have a long chromosome and there's two copies of it, right? And a short chromosome. And we'll say there's two copies of it. They're just floating around in there. Now, during the cell cycle, right, if the cell is going to go through meiosis, first it has to go through G1, and then it has to go through S, right? And so when we go through S phase, remember, each one of those chromosomes develops a sister chromatid. And so now we have duplicated chromosomes, right? And then the homologs pair up on the metaphase plate, right, during meiosis 1, just like we learned in the last unit, or the last lecture, right? So this would be prophase to metaphase 1 from meiosis, okay? And of course then, so forget crossing over right now as we learned. So instead, let's just think about the fact that this cell is eventually going to separate and the homologs are going to go in each direction, right? The homologs separate. And now we have two cells, each with a long and a short chromosome. Okay, but so understand that now each one of these cells is only in possession of a single copy of the genome, right? There's just a single copy of chromosome 1 and 2. And then each one of these cells goes through meiosis 2, so that by the time we're done, each one of these has a long and a short. And I realize I'm writing off the board here, a long and a short. So 1, 2, 3, 4. 4 gametes, each with a long chromosome and a short chromosome. Okay, and so Mendel's factors, as we know today, turn out to be the chromosomes which have genes along them. Okay, so the factors are actually the individual genes. All right, so now let's see, here's DNA. Here's a double-stranded DNA, okay? And let's say that there's just some region of DNA here. Let me draw a little neater here. So here's DNA, okay? And let's say that this region of DNA encodes information to make a protein, some kind of enzyme. So we'll call that region of the DNA, a gene for that enzyme, okay? There can be different versions of this, right? So some people might have a gene that says, say, for example, ATTCCG, and somebody else might have a variant of that gene, say, like ATC GCG, right? So you can see that they differ right here. And so different versions of a gene are called alleles. So you would say, I have the allele for blue eyes, I have the allele for green eyes, etc. Okay? All right, good. So let's now start using some genetic lingo here. So this capital Y versus this lowercase y, okay? So in Mendel's world, for simple factors, you usually name the gene after the dominant trait. In this case, the dominant trait's yellow, and so we'll use a Y. Okay, and the capital letter corresponds to the, therefore, dominant allele. Okay, so that's the yellow allele, right? And then the lowercase letter, that corresponds to the recessive allele. Okay, and so you can see here, let's look at what we're looking at here, the genes of the original true breeding, the true breeding parents. So here's the true breeding yellow and green parents, and you can see here that the reason why they're true breeding is because in their cases they can only donate a single individual allele, right? So what you're seeing here, these represent the gametes. Okay, just two out of the four for meiosis. Okay, and you can see each one is therefore going to have only one letter. Okay, so there's that first law of segregation. Each gamete has a single factor for each trait. Okay, so watch this. We're going to take the pairs of chromosomes, okay, and we're going to now write them out like this. This, therefore, is the genotype, okay? So here, let's just make sure we know the lingo here. So when I say capital Y, or lowercase y, that's the genotype, okay? And because in these cases both of the alleles are the same, we call that homo the same, zygous, homozygous, okay? And because this one has the dominant ones, we call that homozygous dominant, and this is homozygous recessive, okay? So that's two genotypes. And just for our reference here, if you have one of each, that is called, does anybody remember? Right, heterozygous. Okay, you can also call that a hybrid. All right, let's move on. So here is the F1s, okay? And if we think about the kinds of gametes that an F1 can produce, so first of all, let's make sure we are clear on the genotypes. To get to the F1s, we had a capital Y crossed with a lowercase y, right? So the homozygous dominant crossed with the homozygous recessive. So this is the true breeding yellow and the true breeding green. And they come together, and you can see that these can each only donate either a capital Y or a lowercase y. And so when these come together, you have to get this, right? And so this yields a yellow plant. So here's your F1. Okay, so here's your F1. So the kinds of gametes the F1 can make now are a little bit different, right? The 50% chance that a gamete is going to have a green allele, and there's a 50% chance it's going to have a yellow allele. And so if we allow this to self-pollinate, okay, we can take these two options and cross them with these two options. So any possible combination of those are possible, right? And so here are the different possible combinations. If we happen to pick a green one and then another green one, we get a green pea. But you can see that that's only 1/4 or 25%, right? There's only a 25% chance of getting both of these, okay? If instead I grab this green one and then a yellow one, or if I grab this yellow one and then a green one, right? So these two possibles, then what I'm going to do is I'm going to get just yellow. Okay, I'm going to see yellow as a phenotype because remember, this is dominant. Okay, now notice that these are drawn that way. So we have a lowercase and capital and a capital and lowercase. Understand these are equivalent, and we do not ever write things with a lowercase letter first. Okay, so even though these two are different possibilities when it comes to the math, they are equivalent. Okay, they are capital Y, lowercase y. They're both hybrid. Right? In a cell, there's no order this way, right? These are just 3D. In reality, they're three-dimensional representations. And so this is just a way of coding for it, right? So again, so these are the same. All right, so if we can grab a yellow and a yellow, then we would get that. So these are the four possible outcomes of the dihybrid cross. And because there's only a 25% chance of getting two lowercase y's, that's why we only see 25% of the offspring is green. And this is what Mendel observed in his cross. Okay, so there's your 3-to-1 ratio. 3, right? 3-to-1. All right, cool. So when we do genetics problems, okay, we're trying to figure out the outcomes of these. We're trying to predict the future. And we don't always have the ability to examine the genes, obviously. So how do we do this? Okay, so just like in math, genetics has this language that you have to learn to do the problems. So you'll always get some kind of allele key. These are just arbitrary letters that help you keep track of the traits. Okay, so capital letters are dominant and lowercase letters are recessive. All right, so if you see the capital letters together, that's the dominant. If you see the lowercase letters, that's the recessive. Then, of course, if you see them both one of each, you get heterozygous. Okay, so again, while you could technically write lowercase capital, in the genetic nomenclature, we generally write it as the capital then the lowercase, okay? Because you'll understand later that it gets a little hairy and complicated, and so you want to try to keep things consistent. All right, so now we can name things, and you also know how they relate to each other mathematically. Okay. Oh, I'm sorry. You don't know that yet. We also have to know how they relate mathematically. And so what we mean by this is there's two basic laws of probability. You guys learned this when you were kids in elementary school, but let's just really quickly review it. Basically, there's the law of division and the law of addition. I'm sorry, the law of multiplication and the law of addition. So basically, the chance of two independent events happening, two independent events like either heads or tails. So the chances of two independent events happening is the product of their chance occurring separately. So for example, the chances of taking a coin and then landing on heads, and then taking that coin and flipping it and getting another heads, okay, so two consecutive heads, you would multiply the chances of each of those, right? And so the chances of getting a head is just a half, right? And so if you multiply a half by a half, you of course get a quarter, okay? So the chances of getting two heads is 25%. And now if an event can occur in more than one way, okay, so example, you flip a coin, you can either get heads or tails. So the question here is, what are the chances of getting either heads or tails? Okay, then we add the probabilities together. So for example, the chance of getting a head is a half and a tail is a half, and so together that's 1, right, or 100%. So when you flip a coin, you're either going to get a head or a tail. Okay, and yes, you might be thinking, well, sometimes when you flip a coin, you might be able to land it on its side like that. Okay, we're neglecting that idea. Let's say you do it out on a windy day. Never have a chance of doing that. The wind will blow it over and it'll land. Okay, so the probability of a specific event is the number of ways that event can occur out of the total possible outcomes, right? So for example, the possibility of getting one of each, there was a chance of it happening two different ways, right? And it's not that we wrote them left and right. It's the idea that the capital letter can come from the mother and the lowercase can come from the father, or the capital can come from the father and the lowercase can come from the mother, right? That's the idea here. You can come up with, therefore, the hybrid genotype two different ways, and therefore, out of all the possible combinations, 2 out of 4 of those ways, or 1/2, or 1/2, yields that possibility. So that's why we add it, okay? So we can kind of think of it this way now. So here's the beginnings of what we call the Punnett Square. Okay, so let's say that heads is represented by a capital B and tails is by a lowercase b. Okay, and so you can imagine now we have a female and a male that are both heterozygotes. Okay, so what kind of gametes can one of these make? Well, there's a 50% chance you're going to get a capital B, and there's a 50% chance you're going to get a lowercase b, right? And so you can see that here. All right, so what we can do is we can write along the top of a square here the possibilities, and along the sides the possibilities for the other parents. Like this could be mom, and this could be dad. Okay, and then what you do is you look at the different possible offspring, the different possible outcomes. And notice what I did here. Even though it was capital B lowercase capital B lowercase b, right? I still wrote the capital letter first. So even though mom's contributing the capital and dad's contributing the lowercase here, and then in this one, mom's contributing the lowercase, I still write them the same way because that way if I'm counting them, I can see these two things as the same thing. Okay, and then a lowercase b and a lowercase b, we have that, right? So let's just rewrite that nice and neat here, and you can see you have, okay, you have 1/4 of them being true breeding for both homozygous, for both dominant alleles, so homozygous dominant. As a quarter of them homozygous recessive, and then half of them, or 50%, are heterozygotes. Okay, and because we're dealing with a simple trait that has simple dominance, all three of these, okay, the capital B, the capital B, lowercase b, both of them, these are all the dominant phenotype. Okay, and so that's why we would see yellow 3 to 1. Okay. Okay, so let's actually do a real problem now. So in pea plants, we got tall is dominant over short. So because tall is dominant, we're going to use a capital T to represent tall and a lowercase t to represent short. Okay. And yeah, sometimes people get confused with the lowercase letter. It's not T and then S for short. Never do that. This isn't abbreviating from English. This is genetics, right? And so the idea is just we have a letter, and that letter's always got a capital or a lowercase. And the reason why we're using a T instead of an X or an F is because we are doing, we're talking about the phenotype, and it begins with a T. Okay, it doesn't matter what it really is. We're just representing things. All right, so here we go. We got parent 1 is capital T, capital T. Does anybody remember what's the name of that genotype? Right, that would be homozygous. Homozygous dominant. Okay. And then we have the second parent. We're going to call that homozygous recessive. Okay. Now, because we're dealing with diploid organisms, right, they have a pair of chromosomes. That's why they have 2 capital letters. So these 2 capital letters represent 2 chromosomes with a gene where that gene has an allele that we call the capital T allele. Okay. And in this case, this one has 2 genes that have the 2 alleles. I'm sorry, has 1 gene with either with only the recessive allele. Okay. Okay, so they make gametes, and the gametes have 1 chromosome each, and therefore 1 allele each, right? So gametes will only have a single letter for a trait, right? And so the gametes are either T capital or lowercase. All right, so we'll assume mom is capitals and dad's lowercases, right? So mom's gametes can only have 1 kind, dad's can only have 1 kind, and so during fertilization, the offspring get 1 allele from mom and dad, right? And so that's the offspring. That's the, does anybody remember? The F1s, okay? And T is dominant, I'm sorry, capital T is dominant over lowercase t, right? And we know that because in genetics language, the capital letters are dominant. Now, how would you know this before doing the cross? The answer is you don't. These are empirically determined, right? We determine them through the experimentation. So the original experiment, we took a tall plant and we crossed it with a short plant. We had no idea if it even was a dominant recessive relationship. And the outcome was all tall plants. And then when we allowed those to self-pollinate, we got that, right? And so just based on this observation of the patterns of inheritance, we can say that the tall trait is dominant over the short trait. Okay, so all the zygotes carry the allele for tall. Here's the zygotes genotype, right? Heterozygote. They have the same phenotype as mom, but they carry dad's short allele. And so we say their phenotype's tall and their genotype is heterozygous. Okay, and so the Punnett Square is basically mathematical shorthand for thinking about the possibilities that come out of a cross. Okay, and so you can see here is, so by the way, so this isn't a Punnett Square, we're leading up to that. All right, so here's the first cross, here's the parental generation, okay? There's mom's genotype and dad's genotype, okay? And these are the gametes that each of them can make, okay? And then the resulting offspring from this cross is this, right? The first filial generation. This in turn can form these kinds of gametes, right? So this would be one of the F1s and another of the F1s, okay? So we're gonna do a F1 cross, or a, well, that's what, I don't think there's a name for that, just the F1s. And then what the result is, if we think of all the different possible combinations of these, okay, there's only 3 kinds, right? There's this, and there's this. Okay, so those are the 3 different genotypes. And again, remember, the reason why there's 3 and not 4 is because remember that this can be one or the other, capital T, lowercase t, or lowercase t, capital T. Okay, so this is how you set up a Punnett Square. On the top, you put all the possible gametes for mom, and along the side, all the possible gametes for dad. Now again, the possible gametes, the possible genotypes for the gametes, you're not going to put a single letter here for a more complicated situation, as we'll talk about later. So for example, if we're going to deal with 2 traits later, okay, if that's the genotype for 2 traits, the gamete will have 1 letter for each trait, and so here we would write that, okay? You would not ever do this. You see that? Okay. This doesn't represent a letter. This represents a genotype. Okay. And so here we go. The inside of the Punnett Square represents the possible futures or the possible outcomes of the cross. Okay. And so in this case, you can see how many of the what are the chances of getting, say, the recessive phenotype. Right. Just that one, right? And so the answer to that question is 25%. What are the chances of getting an offspring that is a hybrid or a heterozygote? Right. The answer is 50%, right? Or half of them. And then what are the chances of getting a homozygous dominant? The same as the recessive, right? And so 25%. What are the chances of getting the dominant phenotype? Now the answer is 75%, right? We add a half and a half and a half together and we get not a half, a quarter, and a quarter. And so we get 3/4, right? Which is 75%. Okay. All right, so we pretty much just said that. So I'm gonna skip this slide. All right, so now we get to the test cross. The test cross is if we don't know the genotype, right? So you have a tall pea plant, but you're not sure if it's true breeding, so we have to test it out. And it's tall, so it definitely has a T, capital T, but it's the second allele that we're wondering about, right? Is the second allele also a capital T, or is it lowercase t, right? And so we can write this with a T and then just a blank, because we don't know what the second allele is. You could think of that as an X, solve for X. Right, so we do what's called a test cross. We cross the unknown with a known. Right? In order to do that, we need a true breeding recessive strain to do this. Okay? And the reason why we use the recessive trait is because the math works out and it makes it very easy to interpret in that second generation. All right, so for example, we're going to do this cross. We're going to cross capital T, I don't know, cross to lowercase t, lowercase t. Okay, and our job is to find out what this mystery allele is. And the only way to do that is to actually do the cross and then look at the F1 and then ask questions about the F1. Okay, so let's set up the Punnett Square to understand this. Okay, so in one possibility, we have, actually, hold on. In one possibility, so here's the cross. Capital T, I don't know, crossed with lowercase t, lowercase t. Okay, so in one possibility, we have this, right? Why is that just a single one? Why is this more of a rectangle? Because this this parent can only produce a single kind of allele, right? A single kind of gamete. So capital T, lowercase t, and then in this one, we don't know, and then lowercase t. Okay, so let's imagine now, let's run a scenario here. Let's say that the unknown is a capital T. Okay, if that's true, then we can reduce the entire problem to this, right? And so we would say 100% of the offspring would be tall. But let's say that instead it's lowercase t. Then we would have this Punnett square set up. Okay? And we would see that half of the offspring would be tall, but then half of it would be short. Okay? And so by crossing the unknown with the homozygous recessive, if 100% of the F1 was to be tall, that would tell us the original parent was homozygous dominant. If it was 50-50, okay, tall short in the F1, then that would tell us the original parent was, heterozygote, right? Okay, so that's the test cross. All right, I want to take a pause here and then come right back. So, what about more than one trait? Okay, Mendel also wanted to know if dominant and recessive factors for multiple traits either grouped together or didn't. Did the dominant alleles somehow stick together, or were they independent? Right, so for example, we have the capital Y's and the capital T's and the lowercase y's and the lowercase t's. And the question is did these stick together? Did we always get a tall yellow pea plant and a short green, or were we able to get tall green and short yellow? Right, we don't know. So let's go with this. We know the first trait, the pea color, would be capital lowercase y. Okay, and the second trait would be capital lowercase t. And so, did the y's always go with the t's? Did the capital Y's always go with the capital T's, and did the lowercase y's always go with the lowercase t's? Or didn't they? Okay, so to test this, he did what we call a dihybrid cross, which is a cross with strains that are differed in two traits. Okay, so here, this is how you would write that. You would write capital Y, lowercase y, capital T, lowercase t. This would be the genotype of the parent. Okay, so if the dominant factors and the recessive factors always stuck together, then there would be only two possible phenotypes in the F2 generation. And if the factors were distinct, right, if they were independent from each other, then there would be four possible phenotypes. Okay, so again, here, let's show the diagram of this. Okay, so for cross 1, we're going to first start out with true breeding parents. Okay, so tall yellows and short greens. Okay, and so prediction 1, the idea that the dominants stick with the dominants and the recessives stick with the recessives. Okay, what we're going to think of is we're going to imagine that these are the only possible gametes we can make. Okay. Okay, so the first cross here, we're going to start with a true breeding parental generation that is true breeding for two different traits. Okay, so you can see here capital Y, capital Y, capital T, capital T. So this is the genotype to parent 1, and this is the genotype to parent 2. Okay, and you can see here, here's the key, is how do you make the gametes? Okay, oops, so you got this. Now here's how you think of it. The gamete is going to have one factor for each trait, and because there's only one possible combination here, it's always going to be YT. Okay, so these are the gametes genotypes, and because this only has one possible combination, you're only going to get one possible gamete. Okay? Okay. So when you put these together, when you cross these two, you're going to yield a single, so let's watch this, YT crossed with YT. Oops, let me do that again. YT crossed with little yt. You're going to get capital lowercase, capital lowercase. Okay, so there's your F1, 100% yellow tall. Okay, now what we're going to do is we're going to cross the F1 to itself. And so here's Mendel's question. When I have this kind of genotype, okay, so here's a cell that has two factors for each trait, okay, the gametes that are produced, his question was, do the gametes either form only the dominant versions, right? So in other words, does this capital Y trait or factor always go with the capital T, and the lowercase y go with the lowercase t? In which case, if that is true, you would only get two possible gametes. Okay? The alternative is all the different possible combinations of these. Okay? And so think about that. There's going to be many of them, right? Not just two. We can have a lowercase y. Did I just say lowercase y? we can have a capital Y and another capital Y. Okay? And we can have a capital T. Hold on. I'm sorry, guys. I'm tired. I'm going to pause here and restart this. Okay, so if we have this genotype, okay, the question is, what kind of gametes can we make? If the dominant alleles stuck together, as the first hypothesis is, we would only get two possible combinations, right? We would only get the combinations where the capital letters are together and the lowercase letters are together, okay? However, if the dominant and recessive alleles did not stick together, then we can get any possible combination, right? And so we can have a capital Y and a capital T, or we can have a lowercase y and a capital T. Or we can have a capital Y and a lowercase t, or we can have a lowercase y and a lowercase t, right? So there's 4 possible combinations here. So the question was, if we allowed this to hybridize with itself, okay, a dihybrid cross. I'm sorry, if we allowed it to pollinate itself, if we allowed this dihybrid to self-pollinate, okay, which kind of Punnett square would we see as a result of that actual cross? So the 2 possibilities, okay, are either seeing, oh boy, let me redo all this. You got one possibility, okay, is you would see this kind of Punnett square, okay. The other possibility would be, oh, hold on, I'm not done. Let me add on to this. So we would see either this, okay, and so do these crosses. How do you do these? You consider each individual letter at a time. And so capital Y, capital Y, capital T, okay, capital Y, lowercase t, I'm sorry, capital Y, lowercase y, capital T, lowercase t. Okay, and then just follow through here. Okay, so this would be the outcome. So we would say, we would see 25% would be tall yellow, another 25% would be tall yellow, another 25% would be tall yellow. So in other words, we'd see 3 tall yellows to 1 short green. Okay, the alternative is much more complicated. The alternative is any possible combination, right? So capital Y, capital T, lowercase y, capital T, or let's be consistent here, capital Y, capital T, capital Y, lowercase t, lowercase y, capital T, lowercase y, lowercase t. And we cross that, capital Y, capital T, capital Y, lowercase t, lowercase y, I'm sorry, lowercase y, capital T, lowercase y, oops, capital T, sorry about that, lowercase y, lowercase t. Okay, so just to clarify, these are the female gametes, and there's 1, 2, 3, 4 types. And then for the male, 1, 2, 3, 4 types, and they're all the same. Okay, there's the male, there's the female. And so I'm not going to sit here and scribble this out because the next slide has this, but there are 16 possible combinations here. Okay, and when you count up the phenotypes of these, and you do a tally count of, so if you have how many yellow tall plants, how many yellow short, how many green tall, and how many green short. When you add all these up, you get 9, 3, 3, 1. Okay, that would be the phenotypic ratio of a dihybrid cross, assuming both traits have Mendelian patterns of inheritance. Okay, and so sure enough, when he did the experiment, okay, so I just wrote these out. Here are the 2 possible Punnett squares, and it turns out this one was refuted. All experimental data showed that this one was spot on, and this led Mendel to his second law, the law of independent assortment. So for each pair of factors, which we call alleles now, segregate or assort independently from the other pairs. So all possible combinations of factors can occur in gametes. Okay, so an easy way to think of it is that the first law deals with a single trait, and the second law deals with multiple traits. Okay, so to summarize this, what we're going to do is we're going to look at the gametes from the perspective of a representation of the nucleus. So in the middle here, this red box represents a nucleus, and there's 2 chromosomes. Okay, so the first chromosome has the capital Y and the lowercase y, and the second has the capital T and the lowercase t. Okay, so from this animation here, you can see that the lowercase y and the capital Y segregate to 2 different gametes. and then the capital T and the lowercase t segregate to two different gametes, right? And so on the left side, that gamete would have the genotype lowercase y capital T, and the one on the right would have capital Y lowercase t, right? But what if instead the capital Y went to the left and the lowercase y went to the right? Okay, see, that's the key, is that the gametes, I'm sorry, the homologs can segregate independently of each other, right? And so because the first pair can go either way and the second pair can go either way, what that does is it gives us all four of the possibilities, okay? All four of these possible genotypes. So that's what the law of independent assortment tells us. Okay, and so as a result of that, if we do a cross between two individuals that are dihybrid, we can see now we have four different possible genotypes of the gametes for each. And so we can do, you can imagine each one of these has, each possibility has an equal amount of chance of happening. And so as a result of that, there are 16 different possible combinations as represented by the Punnett Square down on the bottom right there. Okay? All right, good. So the only, oh, right, so what you're seeing here, I think this is from your book. On the right here, you're seeing the possibilities if linkage occurred, if the dominant alleles went with the dominant alleles versus if they didn't. So the results on the right show us the segregation is independent. Okay, and so in this example, if you study it, just to show you here, there's two traits being studied here. There's smooth peas versus wrinkled peas, and there's yellow color versus green color, right? So you can have smooth yellow, you can have wrinkled yellow, you can have smooth green, you can have wrinkled green, okay? And wrinkling is recessive, like the green, okay? And so you can see here now, when you do the dihybrid cross, there's your 9 to 3 to 3 to 1 phenotypes. Okay? The only exception to this is when the genes under consideration are on the same chromosome, right? So in this case, when meiosis occurs, the individual chromosomes would carry with them both of the alleles on the chromosome, okay, that are present. So you won't get the lowercase y capital T. Okay, that won't occur because the genes are on the same chromosome. Of course, there's an exception to this we learned about in meiosis 1, which is when crossing over occurs. Okay, and so we'll talk about that in a bit. All right, on to human genetics. The problem with human genetics is that we can't do the same kinds of genetic tests on humans as we can in model organisms like peas or fruit flies or any other of a number of model organisms we use in genetic research. And so instead, we have to use the powers of deductive reasoning to arrive at conclusions. So human genetics is an observational science, not an experimental science. So we're going to go over a few examples of some kinds of inherited disorders in humans that we find. Like Mendel's peas, we can see the same trends in some of these diseases. So for example, there are homozygous recessive diseases where the person would have the disorder if they have two of the recessive alleles inherited. Okay, so instead of thinking of green peas, think that that person would have the disorder. They would have symptoms of the disorder. And likewise, the heterozygote, just like the hybrid pea was yellow, and it was completely yellow, it wasn't partially green. The heterozygote in humans of recessive inherited disorders are usually symptom-free, but they do carry the allele. So here are four examples of recessive disorders in humans. We have albinism, cystic fibrosis, Tay-Sachs disease, and sickle cell disease. Okay, and we'll go through each of these in turn. So in albinism, you have a specific mutation that affects the body's ability to produce melanin. Melanin is a pigment in our skin that gives our skin and eyes and hair their certain colors. There are different kinds of melanin, but if somebody doesn't produce any melanin in certain parts of their body, they will have a very distinct phenotype, which we call albino. And you can see here in this Punnett Square the chances of two parents that are both themselves carriers. Okay, you can see both of them are heterozygotes. The chances of them having a child with albinism is 1/4. right? And so in the picture on the right there, you can see there's a pair of girls, and one of the who are sisters, and one of the sisters is an albino, and the other is not. Cystic fibrosis and Tay-Sachs disease are both very rare genetic disorders characterized by a variety of symptoms. Cystic fibrosis is characterized by the body producing very thick mucus in the lungs and the upper respiratory system, and this is due to a mutation in a chloride transporter that basically prevents the body from producing thin mucus to line the various membranes. And so there's a variety of symptoms associated with this. In Tay-Sachs disease, this is the failure of a certain enzyme within the cell's lysosome that's normally responsible for breaking down certain lipids. And because the cells can't break down these lipids, what ends up happening is they accumulate. And in cells like neurons where they accumulate, this causes the neurons to eventually stop functioning. So individuals who are born with Tay-Sachs disease during the first 2 months of life generally have normal progression, but then after about 2 months, they start to show very severe signs. They show weakening, and eventually it is lethal at a very early age. Sickle cell disease is characterized by a very distinct shape of the red blood cells, only under certain conditions. So in people who are affected by sickle cell disease, they have a tendency to have their blood clot in all the different areas where there are capillary beds. So things like their eyes and their kidneys and their lungs and their all sorts of little areas where there are small capillaries. And it's caused by a single mutation in the hemoglobin beta gene. And what hemoglobin does normally, it fills the red blood cells, and this is basically the carrier that delivers oxygen to our entire body. And when a red blood cell in an individual with sickle cell disease has low levels of oxygen, or actually just average numbers of oxygen, just a little bit, they end up having cells that are shaped in this way. They look kind of like a sickle, right? If you think about a sickle, the farmer's implement, right? That looks like this. They have cells that are shaped like that. And because of the shape of the cell, it causes it to clog into the various arteries. We'll talk more about sickle cell disease when we cover mutations later in the semester. All right, so here's a little thinking question for you. This is called a pedigree. Okay, so pedigree analysis is when we basically look at a family tree superimposed with data related to a genetic disorder. And what we need to do when we look at pedigree is to try to help us understand the patterns of inheritance, which then helps us in diagnosing various diseases. So in this situation, you can see there was a man and a woman who bore two children. Okay, so there's the mother and the father, and you can tell females are circles and males are square. Okay, and an individual who is affected by the disorder is bright yellow. Okay, so here's how it goes. Two people have two female children who are both unaffected. Okay, one of them goes on to marry a man who is also unaffected, and they have a woman who's unaffected. Here's my question to you. How do we know this woman's genotype when we do not know her dad's genotype? I'll let you think about that for a second. So for the red circle, how do we know for certain what her genotype is definitively, as opposed to dad's where we don't know something about him? And the reason we don't know something about dad is because we have no idea who his parents were, if they had it or not. All we know about dad is that he does not have the disease, and the reason we know that is he shows no symptoms of it. Okay, so I'll pause the recording right now and think about it, and I'll give you a minute, and then I'll tell you the details. I'll tell you the answer. Okay, so the answer is, some of you might have very astutely realized she had two daughters that were both affected, right? And the only way somebody can be affected is if they inherit two copies of the bad allele. So they had to get one from mom and one from dad, right? So that's how we know. The key is looking at her children, not her parents. Okay, good. In addition to recessive disorders in humans, there are also dominant disorders. Okay, so a dominant disorder would be one in which the individual who is affected only has to have a single allele to be affected. And four examples of these diseases are Huntington's, achondroplasia, familial hypercholesterolemia, and polydactyly. So in Huntington's disease, this is a disease that usually affects people after the age of 30. Okay, so the key here is that it doesn't impact people until after reproductive age. That's how it gets passed down through the generations. People who do get it end up having some small amount that increases over time of neurodegeneration. And so you can see here two brains, the normal ganglia and the Huntington's disease ganglia, where there has been some very severe damage, particularly in the left lobe in this case. Okay, and so what happens over time is the neurons in the brain, in this diseased brain, end up becoming necrotic, forming huge big holes in the brain, and ultimately that leads to a failure of brain function and death. People who are affected by Huntington's disease vary tremendously, and it has to do with the gene itself. The gene itself has repeat units in it. So the diseased allele is due to these repeating units of DNA. The more repeats you have, the more dramatic the disease is and the earlier the age of onset. So it's possible for some people to have a very mild Huntington's where they don't get it until their 50s or 60s. They only have a few repeats in that gene. However, people who have a tremendous amount of repeats end up getting it much earlier, in their 30s or so, and the progression is much more rapid. Okay, achondroplasia is a form of dwarfism in humans, and it characterized by just having a single faulty allele. Okay, and so you can see here two siblings over here on the left, a normal male and an affected male. And so similar to the other two, like albinism, we have this pattern of expression. Okay. Okay. Familial hypercholesterolemia is, there's not much to say about that other than these are people who, when they do blood testing, have unusually high levels of cholesterol. And the current medical literature tells us now that high cholesterol in individuals, when people have it, it's generally it's genetic. It's not environmental, meaning that having a diet that's low in cholesterol is not going to help them too much. And that's why we have pharmaceuticals like Lipitor to help keep these levels down. So obviously people with high cholesterol levels end up having increased incidence of heart disease and blockages and those sorts of things, strokes, that sort of stuff. Polydactyly is when the individual that's affected ends up with extra fingers or toes. Okay, and so you can see on the left here, you can see the individual has an extra finger budding off of their pinky. There's a tremendous variation in polydactyly. This is just one example. And people who are affected, the phenotype varies tremendously. All right, so in addition to recessive disorders and dominant disorders, there's also disorders inherited through the X chromosome. Okay, so sex-linked inheritance is characterized by genes that are found on the X chromosome and more rarely on the Y chromosome. Okay, so why is this interesting? Well, so think about it. A woman has two X chromosomes, right? So you can imagine if there's a gene on the X chromosome, okay, let's say like the gene is called H. Okay, so a woman will have two copies of it, whereas a man will only have a single copy of it. Okay, so this state is called being hemizygous. Okay, so instead of having a diploid situation or a haploid situation giving rise to a heterozygote or a homozygote, hemizygote means you just have one copy. Okay, and so what's this mean? Well, if there is a disease where, say, a woman has a mutation in it, she's a carrier, right? Similar to the autosomal recessive disorders. So she'll be a carrier, but she herself won't be affected. A male that has the affected allele will always be affected. Okay, and so that's why more men are affected by sex-linked disorders than women, because essentially women have a backup copy, whereas men do not. And incidentally, the Y chromosome has fewer than 100 protein-expressing genes on it. And just for comparison's sake, the typical chromosome has hundreds or thousands. The mammalian XY system isn't the only system in the animal kingdom that determines sex. So for example, in birds, there's actually the ZW system, where you can see in, for example, chickens, the males have the double chromosome, ZZ, where the females have the ZW state. And then in insects, there are actually two systems to talk about. So the first is you can see the XO system in some insects where they have a single X chromosome or two X chromosomes. So females, like mammals, have two X chromosomes, but the male is essentially XO. Whereas in honeybees, for example, there's the diploid haploid system, or the haplodiploid system, in which case the males are haploid and the females are diploid. Pretty cool. Okay, there's three different sex-linked disorders we're going to talk about. One is Duchenne muscular dystrophy, the next is hemophilia, and color blindness. So in all muscular dystrophies, this is essentially characterized by progressive muscle weakening and attrition of muscle cells, the wasting away. And so in Duchenne and other dystrophies like this, they're most predominantly found in males, and they affect skeletal muscles, which are used for movement, and in addition, heart muscles or cardiac tissue. So this, yeah, as I said, this form exists mostly in males. There's actually a really great website of the National Libraries of Medicine where you can go to this website and you can search for all sorts of different genetic disorders and learn about them, including every single one we talk about here. So hemophilia, as most of you probably have heard already, is caused by a defective ability to clot blood. And so somebody who gets cut and they have hemophilia will be unable to staunch the flow of the blood. So there's different kinds. So hemophilia A, in this case, is caused by a gene located on the X chromosome. And it's recessive, right? So that means that if a female is carrying it, so this is what the defective allele is coded as, to distinguish it from B type, I suppose, and the lowercase h indicates it is recessive to the dominant capital H. So we have here an unaffected female whose genotype is XHXH sub A, which indicates that she is a carrier. Here we have a healthy male who has the genotype XHY. And you can see here that there is just like Mendel, there's a 3-to-1 ratio of normal to affected, but when it comes to the male children, right, it's 50-50. When it comes to the females, it's 0. So this is why it's more uncommon. It's less common in females than males. Now, color blindness or color vision deficiency represents a group of conditions that affect the perception of color. And the most common is red-green color vision defects, which is due to, usually there's actually a couple genes involved in this, and 2 of them are involved or located on the X chromosome, giving it therefore more susceptible for males to get it. In addition to mutations that might affect a specific gene, there are also conditions that occur where entire chromosomes are missing, or there's too many chromosomes of one kind, or pieces of chromosomes are damaged or abnormally shaped or missing. And so we'll talk about some of these now. So when you have too many or too few chromosomes, there's 4 conditions we'll talk about here, including Down syndrome, Klinefelter syndrome, XYY condition, and Turner syndrome. This is just a a short list of identified chromosomal abnormalities located in humans. And so you can see here the chromosome that it affects, the actual abnormality, and the associated disease. The ones we're going to talk about that you'll be responsible for understanding are Turner syndrome, Klinefelter syndrome, uh, sometimes we call it double Y syndrome, and then let's see, where is it? Okay, the Cry-Duchat, that's going to be an example of missing a small piece of chromosome 5 in that case. And those are the only ones. Although if you're interested in this sort of thing, or you might know somebody with one of these very rare disorders, feel free to go to that website I talked about earlier and type in these names, and you can learn more. So Down syndrome is characterized by having an extra chromosome 21. And so we call this condition where you have just one extra chromosome of one homolog, trisomy. Okay, so this is different than, say, being triploid. Okay, triploid is when you have every chromosome has an extra copy. Not an extra copy, but just a third copy. There are some instances where this might be normal, right? So a lot of plants are polyploid. And so there might be a plant species that has an extra chromosome or a whole set of chromosomes, right? So you see this, so that would be a triploid. Okay, now of course in humans, this is not a viable genetic combination. Okay, so in the case of Down syndrome, the most common form of Down syndrome, I should say, is when there is an extra chromosome 21. So you would say trisomy 21. Okay, the concept of trisomy, again, it's just one chromosome. So you would say, let's say that there's another situation where you have an extra chromosome 9. You would call it trisomy 9. Okay, I just made that up. There is no, at least I'm not familiar with anything associated with a trisomy for 9. Okay, Klinefelter syndrome is when you have extra X chromosomes. Okay, so in this case, they have, the male here has 2 X chromosomes and then the Y chromosome, excuse me. And this is a very varying disease. In fact, there are some people that are just not even diagnosed when they get it. So it varies in severity. But overall, the symptoms are an abnormal body in terms of proportion. So longer legs than normal, with a shorter trunk, and by trunk, the abdomen area, and abnormally large breasts. Okay, so you'll see an increase in breast size, and in addition, small testicles, and features that are feminine in nature. So for example, the pubic hair actually grows in a pattern that is female. In addition to, and it doesn't say it here, but some of the other websites and books you read, that there's fat deposition is feminine as well. Right, so that's Klinefelter. These people generally also have learning disabilities, and they have varying psychological effects as well. Then there's the XYY condition. Sometimes this is called 47 XYY to indicate the number of total chromosomes. And so you can see here there's 3 sex chromosomes instead of 2. So males that have this condition, right, so they're sexually male, and because they have the Y chromosome, and they tend to be taller than average. And they're, again, like the other syndromes, there's a diversity of severity and symptoms, but on average, these people have an increased risk of learning disabilities with delayed development in speech and language. They have delayed development in muscle coordination, so like a child sitting or standing and walking, and later in life they might be plagued with seizures, having some certain tics, asthma, flat feet, a curved spine, scoliosis, right? But the key here is that it's very, very varied of a condition. And there's also increased risk in behavior, social, and emotional difficulties. Then lastly, there's Turner syndrome. Turner syndrome is characterized by just having a single X chromosome. The phenotype of these individuals is female, and it just so happens this is the only viable monosomy. And what that means is this is the only genetic condition in which there is a single chromosome of the homologous pair, where the human where the zygote can develop relatively normally into a full-fledged adult and have and be fertile and have a viable life. So these females, again, they only have one X chromosome, as you can see in this karyotype. And you can see they also have distinct physical features as well. In general, they have a broad, flat chest, and they have an overly thick neck that has several folds in it. And there's usually associated developmental issues with their toes. They also, similar to the XYY and Klinefelters, have psychological issues as well. Okay, so the last few disorders had to do with having too many or too few entire chromosomes. There's also abnormal chromosome structures. So for example, there's Cry-Duchat and Fragile X. So in Cry-Duchat syndrome, this is when an entire chunk of the small or petite arm of chromosome 5 is missing. And these people, similar to Down syndrome, have distinct facial features. And like the others, there's intellectual disability, there's delayed development. In addition, there's something called microcephaly, which is a smaller than normal head size, weak muscle tone or hypotonia. And the reason why it's actually named Cry-Duchat is because infants that are born with this condition, when they're crying in their crib, sound like a mewing cat. So in French, it's cry of the cat. Fragile X syndrome is characterized by the physical anomaly on the X chromosome, in which it seems that regions of the chromosome are held on by just a teeny tiny thread. This would be, as an analogy, imagine watching a car getting towed and looking closely at it. It's being pulled along by nothing but maybe a few coils of fishing wire, ready to snap at any moment. So these are highly unstable chromosomes, and you can imagine this is going to lead to all sorts of diverse issues. And so it affects males and females, but males are usually more severely affected, again, because they have one X chromosome. There's delayed development of speech and language. There's mild to moderate intellectual disabilities. And these children can have increased higher than average anxiety and hyperactive behavior. And some of them are also on the autistic spectrum. So like Down syndrome and Cry-Duchat and Klinefelter, there's also characteristic physical defects, including a longer than average narrow face, larger ears, a prominent jaw and forehead, unusually flexible fingers, flat feet, like in the previous one, and in males, they have enlarged testicles after puberty. All right, so that's it for Mendel and the basics. So the idea of single traits being controlled by single genes with a pair of chromosomes, a pair of alleles rather, right? So up to this point, we were talking about what are essentially unifactorial genetics. Okay, so let's see. So unifactorial genetics. That's what we've talked about up to this point. Okay, so one gene controls one trait, and that one gene has two alleles where one of those alleles is dominant over the other, which is recessive, right? Okay, so those are the basics. There's a lot of violations of these basics, and we're going to go through them. So the first one is going to be called codominance, and then incomplete dominance, multiple allelic traits, pleiotropy. Okay, and so that's it for unifactorial genetics. And then in multifactorial genetics, which is characterized by the traits being influenced not only by genes, but also the environment, and usually multiple genes. So we'll talk about the concept of polygenic inheritance, or multiple genes controlling a single trait, right? So like that would be one trait is controlled by many genes. Okay, and we'll talk about epistasis, which is when one gene controls another gene. And then of course there's environmental impacts, and we'll talk about that too. All right, so let's get into this. In codominance, Codominance is when you have those two alleles, just like in the pea plants concept with the yellow and green, except instead of one being dominant over the other, both of them are dominant. Both of them express in the adult. And so you can see here, for example, in the camellia flower, you can see a white camellia being crossed with a red camellia, and you don't get just a white or just a red camellia. You actually get a flower that has both of the phenotypes expressed in the adult. Okay, so the key to codominance is both phenotypes are expressed, okay, in the heterozygote. Okay, so if you go back to peas, right, that would be yellow, that would be green, and we learned that that would be yellow. Okay, in the case of codominance, this would have been a pea that, imagine it had, say, like a yin-yang shape where it was half yellow and half green. Then that would have been considered codominant. Another example of codominance is in humans, we have our ABO blood system. Okay, so here's how blood works. You guys know that you have A-type blood or B-type blood or O-type blood or, AB type blood. Okay, don't worry about the RH factor, which is the plus or minus. So A positive, B positive. Forget about the plus and minus for now. All right, let's just focus on the A, B, and O. So this is how we're going to talk about it. There is the capital I and there's the lowercase I alleles, okay? The capital I allele is the functional allele, and what this does is in a red blood cell, let me draw a red blood cell here. So here, capital I, what this does is in a red blood cell, so here's a red blood cell. On the surface of the red blood cell, there's a protein being expressed. And if you remember from one of the earlier chapters, we had, if this is the cell membrane, we had a protein that had some sugars on it, right? That was your glycoprotein, right? Remember, integral membrane proteins. And in this case, these are cell recognition proteins. These allow the immune system to recognize the surface of the cell. This is important for being able to detect other, like viral infections or bacterial infections or any foreign invading protein, any foreign invader. Okay, the lowercase I allele is an allele that does not express anything. Okay, so now you can imagine you have, you get one allele from each parent, right? So you can inherit a capital I or a lowercase I. Now here's the key. The capital I allele can either express the A protein or it can express a B protein, right? So there's two separate alleles here. You either have the A type or you have the B type, or you have both, okay? Or you have the broken one, okay? So you can imagine now for your genotype, you have two slots, right? And those slots can be filled by, oops, let's try that again. You got two slots, and those slots can be filled by either the A, the B, or the nothing, the O. Okay, really, it's not O, by the way. It's really null, like it doesn't do anything. Okay, so imagine this now. You can have either A and A, okay, right? Remember, these are all with the I's here. So let's just to keep it simple though. You can have either AA, you can have AB, or you can have AO. Okay, you can have BA, which would be the equivalent of AB. Those are the same, right? You can have BA. You can have BO, which is the smelly one. All right, get it? BO. Okay, and then you can have OO. All right, so those are your genotypes. Now let's think about your phenotype. If you have A type, if you have AA, you would have the A phenotype. AB would give you the AB phenotype, but what about this one? That would also be A. And what about this one? That's also AB. Right, those are the same. And what about this one? That's B. And then this one is O. Okay, so these are your different blood types. And let's just reduce that. You can have either A, you can have AB, which is the same as this one, or B, or O. Okay, so that's how blood type works. So the key here is that if you are AB type, you express both proteins like you can see here. Okay, so your immune system knows and sees the A protein and the B protein. And so you're able to accept either of those if you get a blood transfusion. Okay. Now, as for the Rh factor, you have Rh plus, Rh, or Rh minus. This is a separate gene, okay? And you either have the allele that makes it or you don't, okay? And so now you have A-type blood that has the Rh factor, or you have the A-type blood that doesn't, okay? And so if we think about, on average, the AB positive, okay, that's the universal acceptor, okay? A person with AB positive blood can receive A blood or B blood or AB blood that's either plus or minus, okay? And so they can accept any of it, or of course, O negative or O positive, right? They can get any of those. Their immune system knows it all. In contrast, the O negative person, they can't receive any other blood except O negative, right? However, they are the universal donor. Universal donor, because their blood can go into any other blood. That's why they're so important in the population. And that's why if you have O negative blood, it's very important that you make sure that you donate your blood. If you can, if you're healthy enough and if you're okay with that. All right, as for, they talk about antigens. All an antigen is simply something that you can think of as stimulates the immune system. Okay, and so it's generally a small polypeptide. It's a piece of a protein. Okay, usually about maybe 8 to 12 amino acids long. That's all an antigen is. Okay, and so, or rather, you know what? I just defined something called an epitope. I know, I'm sorry, I'm getting into the weeds here, but just to clarify, an epitope is a small polypeptide, okay? Antigens are just anything that stimulates the immune system. But when you dig into the antigen, you always find something that is an epitope. So, for example, we say that we're allergic to cats, okay? So if you're allergic to a cat, let's just draw a silly little cat here. Okay, there's our cat. If you're allergic to a cat, the reason you're allergic to the cat is because his skin cells are shedding. Okay, and if you dig into a skin cell, right, here's a skin cell, there's a protein on the surface of that skin cell that has an epitope on it, and that epitope is what causes your immune system to go, oh, this is really bad, and you start sneezing and your eyes start watering, and this might lead you to develop a hatred for kitty cats. All right, so here, let's do a problem. I'm going to make you do this. So let's say a man with blood type A is planning on having a family with a woman whose blood type is O. And he's blood type A. When it says heterozygous, I want you to think about what that means. What does it mean by having A-type blood but is heterozygous? Okay, all right, so what are the genotypes of the man and the woman? And then go ahead and do the cross. All right, so pause the video and try to work through this, and I'll give you a couple seconds. Okay, so the man has type A blood, A-type blood, but it's heterozygous, which means he's got to have some other allele that's not A. The only other allele that can give you A-type blood and not be A is the O allele, okay? Whereas the woman who's O-type, okay, so this is the male, the woman who's O-type has to have double O's, right? So it's a pretty simple cross to set up now. You can either generate a gamete with an A or an O, okay, and then the gametes for the female, she can only make an O. Right, I'll just do this to make it very clear that we're dealing with an O or a null. Okay, so do the cross, and what are the chances? Okay, so you either have AA, or rather AO or OO. So the children have a 50% chance of having A-type blood and a 50% chance of having O-type blood. All right, great. So that was all codominance. Incomplete dominance, which gives the phenotype that we would see in blending, is when the heterozygote has an intermediate phenotype. Okay, so there's three phenotypes. So for example, here in this particular flower, we have a red flower and we have a white flower. And when you allow those to hybridize or cross-pollinate, you get a pink flower, just like in blending. Okay, but let's play this out. Let's imagine that the alleles stand for like faucets. Okay, so you have two faucets, and the faucets are either on or they're off. Okay, and so you can imagine if you have two faucets, I don't know why that keeps disappearing. If you have two faucets that are both on, you're going to get a nice deep red. Okay, so R1 would signify on. And then R2 would signify broken faucets, faucets that don't generate any pigment. Okay, and so if you're R2, you have white. Okay, so you can imagine getting one good faucet and one bad faucet, you're going to have just a little bit of paint, so to speak. And so you'll be a lighter red or pink. Okay, but now play this out, right? So the gametes that you can make here, right? Well, there's only one kind of gamete. Oh, hold on. Let's play this out. Let's do the monohybrid cross. Okay, so you can either contribute an R1 or an R2 to the F1 generation, right? So this cell can make either this gamete or that gamete. Okay, and this one or that one. So here's your egg, and here's your sperm. And so you can see here, R1, you get red. R2, R1, you get pink. R1, R2, you get pink. And R2, R2, you get white. So just like in Mendel, if you look at the genotypes, I'm sorry, I'm not going to do those three. So here's the big difference. Okay, there's three phenotypes instead of two. There's the red, the pink, and the white. Okay, and now you can also imagine the fact that if this was blending and you cross pink to pink, you would always get pink forever, right? And so this is a really good example of why blending isn't true, right? We're mixing two pink flowers and we're 25% of the time getting red and the other 25% of the time getting white, right? Just as white as grandpa and just as red as grandma. So you can understand now that even though some things look like they're the blending of their parents, in reality, they're still inheriting discrete factors. Epistasis. Epistasis, which is Latin for standing upon, you can think of the idea here is one gene standing on the other, preventing it from doing its job. All right, so in this case, we have a chromosome with two genes on it, okay? And so you have the functional gene A, and if gene A is functional, what it's actually going to do is suppress gene B. Don't worry about the mechanism of how it suppresses it. Those can vary dramatically, but for one example, you can imagine this gene encodes for some kind of protein that then literally goes and binds to this gene and prevents it from doing its job. Okay, and so if you inherit a broken A gene, like for example on this chromosome, you can see that in the absence of a functional gene A, gene B is on. Okay. So epistasis or epistatic gene control generally talks about the influence of one gene on another in terms of shutting it off. Of course, there are exceptions to that. We're going to talk about one really fun example here. So this is Labrador Retriever coat color. This is how it works. There's two genes involved that help decide whether the dog is a chocolate, a yellow lab, or a black lab. And more recently, people are breeding and understanding red labs. This is not going to help you explain red labs. I still don't fully understand the genetics of red labs, but I will after I read more about it. I learned about it recently. So anyway, there's two genes. The first gene we'll call the B gene. Okay, so there's B, capital B or lowercase b. And the first gene, therefore, does basics in coat color. It allows the expression of either a black pigment or a brown pigment. And brown is recessive and black is dominant. Okay, so if you have either of these two genotypes, capital B, or capital B, lowercase b, you're going to have a dog with a black coat. And the only way to get a brown coat is to have lowercase b, lowercase b. Okay, now there's a second gene, the E gene, And what you can imagine the E gene's role is to take that pigment and then deposit it in the hair, right? So if this, excuse me, if this, oh boy, here we go. If this is a hair cell, okay, you can imagine the pigments are made in the cell, and then there's a gene that makes an enzyme that allows those pigments to move up and into the hair, and so you have hair that's a certain color. Okay, so you can imagine the E gene is creating a little machine that allows the pigments to be stuck into the hair. And you only need a single functional allele to get it to work. Okay, which means as long as you get a capital E, you will have a color, whether it's black or brown. Okay, if you inherit 2 copies of the recessive allele for the E gene, then you get no color. And if you get no color, that expresses as yellow. Okay, so yellow labs actually have the genes to make black or brown, but they're just not showing it. Okay, so here's how it works. If you have capital B, or capital B, lowercase b, okay, or lowercase, lowercase, but you have 2 lowercase e's, all of these dogs are yellow. It doesn't matter what these are because you can't deliver the pigment to the hair. Okay, however, if you have capital B, or capital B, lowercase b, and you have just a single E, okay, whether it's, okay, so we have capital B, capital B, capital B, capital E, or capital B, capital B, capital E, lowercase e, it doesn't matter. As long as we have one of those capital E's, both of these will be what color do you think? Right, those would be black. And what about if you have capital B, lowercase b, capital E, lowercase e, so like a dihybrid? What color would that be? Right, black, because it's right here, right? Black's dominant over brown, and you have the capital E. Okay, so if you look over to the right here, the Punnett Square, you can see a setup similar to our dihybrid. If you remember our dihybrid, that would be the capital Y, capital T, capital Y, capital T, lowercase y, right, crossed with itself. It's similar to that. Okay, except the key here is that the phenotypic ratio is not 9 to 3 to 3 to 1. Okay, in this case, it's actually 9 to 3 to 4. Okay, so you can see here are your yellow labs. You can see the reason they're yellow is because they're all lowercase e's, even though they encode the genes to make black or brown. Okay, so that's basics of Labrador breeding. Now there's polygenic inheritance, which means you have 2 or more genes that contribute to a single trait's appearance. And a really simple example of this is your skin color. So we've identified to date about 8 genes that are responsible for skin tone. And you can see here a sampling of those. So there's 6 in this case, sorry. So each one of these dots represents either the on allele or the off allele. Okay, and so if it's black, what that means is you have the allele to make a lot of melanin. And if it's white, it means you don't. Okay, so you can see here, this person and this person each have 3 ons and 3 offs. So they have a middling skin tone. And if you put these 2 together, right, and they kind of look basically like a trihybrid. We don't want to do that Punnett Square, that'd be scary, right? But if you did do it, you'd see that there's a 1/64th chance of having the lightest of light, and equally a 1/64th chance of the darkest of dark. Okay, and so these probabilities fall on basically a standard curve or a bell curve, a normalized distribution, as they say. Okay, and so here's just another example of here's a Punnett Square, and you can see we took the 6. So this is basically a trihybrid cross. Okay, and so you can see there's a total of 64 squares, and you can see the spectrum of skin tone. And in reality, it's not 6 genes. Oh, I'm sorry. You know what? This is just showing you 3 genes, not 6 genes. There's 6 alleles. Okay, so in reality, there's 1, 2, 3, 4, 5, 6, 7, 8. Okay, that would be an octahybrid cross, and we're not doing that. But that's the gradation of human skin tone. Okay? And so you can see now that's why all of our skins are so varied from each other. Simply there's so many different combinations of those 8. But what's neat is that every population on Earth, every human population on Earth has those same 8 genes and the same combinations of those alleles. So in reality, what we see as differences between people because of their skin colors, it's only due to 8 genes. And so that concept of race is not really a biological term. It's more of a sociology term. Biologists don't acknowledge the notion of race. Races are not a thing in biology. And so it's like the difference between, I don't know, a pair of, mini schnauzers that have, black hair or gray hair. Like I have a pair of dogs, Buttercup and Wesley, and both of them are litter mates. And they're mini schnauzers, and they have black hair. Wesley has black hair, and Buttercup has gray hair. So the differences that we see in outward appearance between people is literally just skin deep. Okay, so environment also plays a huge role in phenotypic appearance for a lot of traits. So for example, in flamingos, the determination of whether or not they have those really beautiful deep pink or orange feathers has really to do with their diet. So they eat shrimp, and when they eat the shrimp, those shrimp have a lot of pigments in their exoskeleton. And when the birds digest it, a lot of those pigments end up finding themselves being stored in adipose tissue, which then lead to feather development. And so you can see that if you take a flamingo and deprive it of a diet of colorful shellfish, it will eventually be white. Okay, in contrast, or in addition, you have hydrangeas, right? So we all see hydrangeas in the springtime here. Up in the north, we mostly see red hydrangeas, and that's because the soil up here in the north is generally alkaline. Okay, so the color has nothing to do with the genes and everything to do with the pH of the soil. In the south, where the soil is more acidic, hydrangeas are blue. Okay, so these could be the same plant transplanted in different pHs and blossoming in different seasons, or rather different years, and they would yield completely different colors. Okay, now there are some animals that have what are called temperature sensitive mutations. So in the example here, we have rabbits that are reared either in really cold or really warm environments. And you can see for the one that was raised in a cold environment, only in the tippy extremities of its body, where if you could think about it, in the wintertime, where do you get cold first? Well, your ears get cold, and your nose gets cold, and your hands get cold. And I don't know about our feet are usually covered in boots and socks, so we won't count that. But the idea here is that these ends or edges of our skin are the coolest areas. So a bunny raised in the cold and having pigmentation in the very tippy tops of all of its extremities, that shows us there's an enzyme that's being expressed that doesn't work at high temperatures, but it does work at low temperatures. Okay, so this isn't genetic. This is environmental. And lastly, if we take a pair of twins, people who are genetic clones of each other, and we raise them in 2 different environments. One is raised in an environment that is not healthy and is malnutrition when they're growing up. The other is fed a good diet. You can see that there's a clear difference in height. Okay, so our height is not just controlled by our genes, but also by the diet we had when we were younger and growing. All right, and last but not least, we have what's called pleiotropy. Pleiotropy is the opposite of polygenic inheritance, where instead of one trait being controlled by several genes, we have one gene controlling several traits. Okay, and a classic example of this is the disease phenylketonuria, or PKU. This is due to mutations in the PA gene, PAH, and what this does is it mutates an enzyme whose job is to normally take the amino acid phenylalanine and convert it to tyrosine, another amino acid. And for people who can't do this conversion, this ends up building up in their blood and going places it's not supposed to, leading to all sorts of problems. The good news is when somebody is born and screened for PKU and found to have the mutation for it, if they're raised on a relatively strict diet that does not contain too much phenylalanine, then they will have a relatively normal life. Okay. Otherwise, if they're subjected or exposed to phenylalanine especially during childhood, early childhood, they get all sorts of problems, including mental retardation, seizures, behavioral problems. And what's most interesting is this musty or mouse-like odor, and that's due to the buildup actually of phenylalanine on their skin. So they actually have a certain smell to them. They also have other skin problems like eczema and pigmentation abnormalities. Okay, if you've ever looked at like a can of diet soda and you look on the label, you might see something that says something like pharmacokinetics, warning, contains phenylalanine. Okay, and those labels are there to prevent these people from accidentally ingesting too much phenylalanine. Usually you'll see it in anything diet related that has NutraSweet, because NutraSweet is actually a phenylalanine derivative. Okay, so that's it for simple genetics, I should say. Okay, so I'm going to take a little break here and come right back, and we're going to finish this chapter up.